Namaskar. Today we are going to discuss the difference between antigen and antibody. We know antigen and antibody are part of the immune system. The antigen is foreign to our body and antibody is endogenous. Our body produces. So if we discuss the definition, the antigens are the foreign particles that are capable of triggering an immune response, and usually they are harmful substances. They cause infections, they cause diseases, they cause allergy, whereas antibodies are the special molecules produced by B cells, mainly the plasma B cells of the immune system in response to the antigen. So we can say that antigens are harmful substances and antibodies are the protective substances produced in response to antigen. So antigens are foreign particles. They are, when introduced into our body produces antibody as a protective mechanism to eliminate that antigen. So antigens are also called as immunogens and antibodies are also called as immunoglobulins. Antigens, immunogens, antibodies, immunoglobulins. So you can see here, suppose a bacteria, it can cause infection. So on its surface there are epitopes, and antibody are Y-shaped glycoproteins, and on their surface there is paratope to which the antigen actually binds. So the epitope of antigen binds with paratope of antibody. So antibody is the Y-shaped structure and it has two heavy chains. This is one heavy chain, this is another heavy chain, and two light chains. So this is the structure of antibody. So any pathogen on its surface has epitopes to which the antibody binds, and the part of that antibody which binds with the antigen is called paratope. So these differences you can write that specific binding site in antigen is called epitope and specific binding site in antibody is called paratope. So epitope is the interacting domain of antigen with the antibody, whereas paratope is that which recognizes a specific epitope on an antigen. So mostly the antibodies are glycoproteins, whereas the antigens, though mostly they are proteins, they can also be carbohydrates, lipids, as well as nucleic acids. And as we have discussed that the antigens can be bacteria, virus, fungus, protozoa, all the infectious agents. So they can be cells, the antigens can be cells, but the antibodies are not cells. This is one difference. So you can see here, this is a bacterial cell, and on its surface there are epitopes, and to this epitope the antibodies bind. This Y-shaped structure is called antibody, which binds to the epitopes of the bacteria or the antigen. So antigens cause diseases, infections, allergic reactions, whereas the antibodies are protective and they protect our body from the antigens by either immobilizing the antigen or by lysing the pathogen. So you can see here antigen and antibody reaction. So if we see the structure, the antibodies are Y-shaped structure containing 4 polypeptide chains, 2 heavy chains and 2 light chains, as we have discussed. 2 heavy chains and 2, these are 2 heavy chains and these are the 2 light chains. Whereas if you see the structure of antigen, it is complex and variable and depends on the type of pathogen. So the epitope present on the surface of antigen reacts with the paratope on the surface of antibody. And if you see that suppose a lock and key pairing, so one acts as a lock and other acts as a key. You can see here epitope in the antigen and paratope on the surface of antibody. So the antigen acts as the key, and the antibody acts as the lock. So you can see the structure here. The antibody acts as the lock and the antigen acts as the key. So these antigens are present on the surface of microorganisms or as free molecules, whereas the antibodies are present on the surface of plasma B cells or in the body fluids. So in antigen-antibody reaction, antigen acts as key and antibody acts as lock. There are 2 types of antigens. Mostly they are foreign substances, but sometimes we can have autoantigens. That means our immune system is activated against our own body parts. For example, in case of rheumatoid arthritis. In rheumatoid arthritis, the antibodies are developed against the synovial joint, and the antibodies are of 5 types. Antibodies or immunoglobulins are of 5 types based on the type of the heavy chain, the constant region of the heavy chain. They can be immunoglobulin A, immunoglobulin D, immunoglobulin E, immunoglobulin G and immunoglobulin M. Then the preparations of antigens are called as vaccines. So we usually take the vaccines which are mostly the pathogenic character is attenuated, and the preparations, these vaccines we usually take so that our immune system gets activated, and when actual antigen comes or actual infectious agent comes, our immune system is prepared. Our immune system has already the antibodies to tackle with those antigens. And the preparations of antibodies are called as sera or anti-sera, and these preparations we usually give when immediate response is required. So we can administer a preparation of antibody to eliminate the antigen. So for quick response we usually give the preparation of antibody, and the preparation of antibodies are known as sera and anti-sera. The antigens they bind usually to a macrophage to reach a helper T cell to initiate an immune response, whereas the antibodies they react with antigen directly to destroy them. And the antigens are usually processed in the macrophage and sometimes in the dendritic cells. So we call that as the antigen presenting cell, APC, and that will be recognized by the helper T cell, and then the immune response is, the specific immune response is initiated. So for diagnosis, the test for diagnosis, we can detect antigen, also we can have test for antibody to diagnose any disease. Usually the antigen tests for diagnosis are cheaper, quicker or faster, and they usually detect the presence of the infectious agent or the antigen. Whereas the antibody tests for diagnosis are not quick, they require extensive knowledge on clinical equipment, and usually they detect the presence of antibody, whether our body has developed antibody in response to the antigen or not, and whether they are present in the blood or serum. So this is all about the difference between antigen and antibody. I hope this will be useful to you. Thank you.